Ever since man settled down in a place to start a social and cultural life, he began cultivating crops. Nearly all the present-day crop plants were developed by prehistoric man by altering the wild ancestral species. The present-day plants are improved varieties and have been produced by careful artificial selection and plant breeding practices. Thus, the science of improvement of plants by improving their genetic potentialities is called plant breeding. There are many techniques involved in plant breeding. Hybridization technique. It is a technique of plant breeding in which the two plants having desired characters, that is, those plants which are genetically dissimilar, are made to cross and develop seeds. The pollen grains of one plant selected as male are collected for pollination. The stigma of another plant selected as female is dusted with the pollen grains of male plants. After pollination, the fusion of desired male and female gametes result in hybrid embryos having characters of both the plants. The seeds of these plants are collected and grown in the fields. There are various types of hybridization. They are intervarietal hybridization. It is the cross between two plants of the same variety. Interspecific hybridization. It is the cross between two different species of the same genus. Intergeneric hybridization. It is the cross between two different genera of the same family. The second important technique in plant breeding is genetic engineering or recombinant DNA technology. Methods like selection and hybridization involve the complete genome of the plants. The latest recombinant DNA technology involves transfer of one or more genes from one plant to another. The plant in which foreign gene has been introduced is called transgenic plant or genetically modified organism or GMO. Aims and objectives of plant breeding. The various factors for which variety improvement is done are as follows. High yield. It is the main concern of the plant breeder, which is achieved by developing and selecting more efficient genotypes to increase the productivity. Improved quality. Quality is another objective of plant breeding. For example, sweet, juicy and seedless oranges are preferred over a large number of sour ones. Quality improvement is required in grain size, protein quality in pulses, cooking quality in rice, malting quality of barley, etc. Biotic and abiotic resistance. Under natural condition, the crop plants are prone to certain biotic and abiotic stresses, which causes a great loss of production. Biotic stress is the diseases caused by bacteria, fungi, etc. or damages caused by insects. Abiotic stresses include drought, salinity, heat, etc. The biotic and abiotic stresses can be tolerated by newly generated crop varieties through genetic manipulation of the DNA. Changes in maturity duration. By reducing the time duration from sowing to harvesting, the production of a crop can be increased many times. The farmers can grow many rounds of crops in a year in the same field by reducing the duration of crop. Uniform maturing of the crop makes the process of harvesting easy and reduces losses during harvesting. It also reduces the productivity cost. Wider adaptability. Ability to withstand extremes of moisture, drought, temperature, etc. by cultivated plants is another desirable trait required for breeders. Developing such varieties which can be grown under different environmental conditions help farmers to grow one variety at different climatic conditions in different areas. Desirable agronomic characteristics. Plant breeders have also extended their hands towards the improvement of plants for other needs. For example, cotton with long fibers and more fabric strength has been developed through hybridization.